In 2002, a gene turned up broken in two identical twins. Both had autism. So they named it for autism itself, Autism Susceptibility Candidate 2. But follow that same gene to the other children who carry it, and the name starts to slip. Most of them aren't autistic at all. The gene sits on the 7th chromosome, and it is enormous. More than 1 million letters of DNA, one of the largest genes we carry. 19 pieces, switched on hardest while the brain is still being built. Its name is AUTS2. Nobody was looking for it. Two identical twins came in with autism, and a picture of their chromosomes showed a break. A piece of chromosome 7 traded onto chromosome 20. The break cut straight through a gene no one had named. 5 years later, the same gene turned up broken again, this time in children with intellectual disability and no autism at all. The gene was doing more than its name promised. How do you catch a broken gene this big? For years, one test did the work, the chromosomal microarray, reading a whole genome from missing pieces. It is how most people with AUTS2 were first found. A deletion, whole exons simply gone. But the microarray is blind to the smallest errors. A single changed letter slips right past it. Now, sequencing reads the gene letter by letter, and those hidden variants surface too. So what does the gene actually do? Deep in a young neuron, its protein takes a seat on the DNA, joining a complex whose usual job is to hold genes shut. Polycomb, the silencer. But when AUTS2 joins, the switch flips. The complex that kept genes off now turns them on. And the genes it wakes are the ones that build a brain. But the protein doesn't stay in the nucleus. Out in the body of the cell, it works as a guide, steering newborn neurons to their place in the cortex and pushing out the branches that will wire them together. All of it timed to the moment the brain takes shape. 2 working copies of the gene run this program. Lose one, and there isn't enough to finish the job. In 2013, a scan of nearly 50,000 patients gathered the scattered cases into one diagnosis. The picture is consistent. Development runs slow. Speech comes late or never. The head grows a little small. Feeding is hard from the start. And yet, again and again, the same note in the file. These children are worn, sociable, quick to smile. It is rare. Around 60 have been described in the medical literature. Almost certainly, there are many more. There is no drug for AUTS2, no edit that puts the missing copy back. What helps is time, used well. Speech therapy, occupational therapy, physical therapy, care aimed at each symptom as it appears. None of it targets the gene, but begun early, while the young brain is still plastic, it can bend the whole course of a childhood. Which is why the diagnosis matters. It opens the door to that help sooner. In the lab, the work goes on. Zebrafish and mice missing the gene, mapping what it builds and what breaks without it. From 2 twins to a syndrome with a name. From a name about autism to a gene that helps build the brain. We know what it does now. We're still learning how to help. The Gene Channel.